So, hi everybody. It's good to see so many people here, except the speakers. Okay, it's good to see them as well. Anyway, so welcome to this session on genomic and molecular visualization. My name is Michael Krone. I'm from the University of Tübingen in Germany and New York University. And we will have a series of exciting talks. I'm very much looking forward to that. We will start with Sehi Lee, multi-view design patterns and responsive visualization for genomics data. So, stage is yours. Oh, and people, please ask questions in the Slido.
Hi, everyone. I'm Sehi Lee. I'm a postdoctoral research fellow from Harvard Medical School. In this presentation, I will introduce our study on multi-view design patterns and responsive visualization for genomics data. Visualizations are heavily used in the genomics data analysis. There are hundreds of different visualization tools in the wild, which explain its essential role. One of the typical examples of using visualization in the analysis pipeline is interactive data portals. Genomics data sets are commonly shared through public websites, and oftentimes interactive visualizations are embedded for seamless data analysis. For designing such websites, one of the key aspects to consider is responsive design, which means you need to think about supporting varying screen resolutions for your visualization. A project manager from our interview highlights the importance of this problem by saying that in many hospitals, screens are very old, so a front-end engineer always think of the responsive aspect of the website. In this work, we wanted to understand how we can support smaller screens in genomics data visualization, and more specifically, considering the nature of multi-view genomics data visualization, we wanted to find a way to design responsive multi-view genomics data visualization. To answer this question, we followed this study process. First, we surveyed existing multi-view genomics visualization to identify to what extent they support responsive designs. Also, we wanted to identify view composition patterns as well as their usability issues in smaller screens to better understand current challenges. Based on the survey result, we identified several high-level approaches for responsive genomics visualization. And lastly, we extended existing visualization grammar for responsive designs. So from the survey, we reviewed a total of 40 different web-based tools. We tested individual tools in 5 different logical resolutions taken from existing research. That includes desktop, tablet, and smartphone screen resolutions. Here, I want to summarize these 3 main findings from our survey. First, genomics data visualization uses many number of views at once. On average, 8.1 views are shown when a user opens a tool for the first time. And this number is larger than a survey result outside genomics, where the author found that most of the multi-view visualizations showed less than 5 views at once. Furthermore, majority of the tools allowed adding unlimited number of views, which means in theory, genomics users use many number of views at once. These increases the difficulty for designing usable responsive visualization due to the screen scalability. Secondly, we found that 4 view composition patterns are the most common ones that cover over than 90% of the all visualization. That include vertically long composition that uses vertical juxtaposition only to compose views, horizontally wide composition that mainly uses horizontal juxtaposition, circular view-based composition that incorporate any of the radial visualization, which is quite common in genomics. And lastly, cross-shaped composition that uses matrix visualization and juxtapose views on 2 or 4 directions on top of the matrix. The vertical composition was the most popular one in our survey that covers 70%. And interestingly, again, this tendency was different from visualizations outside genomics, where their survey showed that horizontal juxtaposition was the most popular one. The last key insight is that existing visualization tools supported limited responsive designs. Most of the responsive designs we found are quite simple ones, like changing width and height in smaller screens, and none of the tools incorporated any of the multi-view design aspects in smaller screens. And the lack of responsive designs resulted in different types of usability issues. These 5 charts represent the number of usability issues we found when tested in 5 different screen resolutions. And you can see that majority of the tools have usability issues when the screen size decreases. A typical example is readability issues, where our visualization becomes too small to interpret. And another typical example is out of viewport issues, where our visualization becomes impossible to see at once. This makes users to constantly scroll the page, reducing the effectiveness of the visualization. So through the survey, we identified the composition pattern as well as usability issues and analytic tasks that stem from the composition patterns. And based on this, we reviewed literature based on related to multi-view design, responsive design, and genomics visualization. And we identified 5 high-level approaches that can address observed usability issues. For each high-level approach, I want to share 1 or 2 examples in this presentation. First approach is changing arrangement in different screen resolutions. One of the issues with vertically long composition is that users have to scroll up and down frequently to compare between 2 sets of views. And in an extreme case, one comparative tool in our survey showed as much as 52 views at once that are stacked vertically, meaning that users have to scroll for a long distance to compare between pairs of views. To support such a pairwise comparison, one can instead directly juxtapose pairs of views so that the comparison can be more effective. Another common issue is when browsing many metrics views at once in smaller screens. To overcome the screen scalability issue, these matrices can be changed to a novel design called complementary matrix, which is known to be as effective as using the full square for individual matrices while reducing the use of screen spaces. Another approach is changing the layout of the visualization. Common issues with the circular visualization is that it becomes too small in mobile screen resolutions due to the skewed aspect ratio of the screens. To save spaces, this multiple bar chart can be changed to stacked bar charts, which can be eventually changed to linear layout so that it uses a screen space more efficiently in smaller screens. Last 2 examples are overlaying guidelines and shortening distance. Genomics visualizations are commonly viewed with reference genes, as shown on the top of this vertically long visualization. And users often try to find correlation between this view and then the rest of the views. However, views on the top is far from the reference view on the top, so the users have to rely on their working memory to find correlation. To support more effective one-to-many tasks, one can overlay such the important information directly on the individual view, which is shown as a dimmed background of the visualization, or pin the view on the top of the screen so that users can interactively shorten distance between reference view and then target view for better comparison. In the grammar, we enabled responsive designs by allowing users to define sets of logical conditions based on screen resolutions and aspect ratios, and users can also define corresponding alternative specs. So depending on the logical condition, the original specification or the alternative ones can be used for rendering visualization depending on the screen resolutions. In our paper, we provided a full table that lists multiple approaches by composition patterns, usability issues, and task types, so that designer can more easily find the potential responsive designs for their visualization. And also, adopting the idea of text variant visualization, we provided more in-depth visual summary of the view composition patterns. In summary, our survey revealed that we need more genomics visualization that support responsive designs. We found 4 typical view composition patterns which can be used as a composition taxonomy, and many number of views are used at once in genomics visualization, which makes the support of responsive visualization really important, but at the same time challenging. As responsive designs, we identified 5 high-level approaches based on literature review, and our preliminary user study shows that the configuring logical conditions and alternative specs in the extended grammar are simple enough to understand, while the dependency to the original grammar would make it difficult for novice grammar users to use the responsive design before getting familiar with the grammar. If you're interested in learning more, please check out our public interactive demo websites. And with that, I would like to end my presentation. Thank you for listening.
Thank you for this interesting presentation. We have time for one quick question. So David Corrill asks, I know you mentioned in the paper that looking at interaction was beyond this paper, but do you have any ideas how interaction could play into the responsiveness?
Yeah, I think interaction is also a really important part when you think about the responsive design. And in the existing paper that surveyed the existing responsive design and also come up with the taxonomy, interaction is actually an important part. In our survey, we focus on the visual representation as well as the multi-view design aspects when considering different screen resolutions. And I think that could be one of the that is one of our limitation, but I think that is important aspect that we need to conduct a follow-up study. I think especially using the input widget in the websites, or even incorporating input modality as a main factor is important part. In the mobile devices, touch interaction is also really important.
Okay, thank you. So I guess in the interest of time, we have to move on. So let's thank Sei again. And the next talk will be a virtual presentation by David Kutak, Vivern, virtual environment for multi-scale visualization and modeling of DNA nanostructures. And of course, he is here later live for Q&A.
Hello, welcome to this talk. My name is David Kutak, and I will present our paper Vivern, a virtual environment for multi-scale visualization and modeling of DNA nanostructures. Before doing so, let me first remove my headset and then express my thanks to all of the co-authors involved in this paper. Also, let me express my thanks to all the institutions supporting our research. Before diving into the main topic of the presentation, let me introduce you to the DNA nanostructures. As the name says, these are nanoscale small structures created from DNA, and their construction relies on the predictability of DNA base pairing. Despite the fact that this field is still rather new, it already managed to reach various achievements. Since the design of such structures is becoming increasingly complex, this field can heavily benefit from novel computer-aided design techniques, making it easier to model, explore, and evaluate the structural designs before performing the transition to reality. So as for the size, for example, on the left you can see a design of a nanostructure having tens of nanometers, and on the right image you can see atomic force microscopy of the same structure. And with additional computer-aided design tools, we can make this transition much faster and also much cheaper. Among the paradigms for the design of DNA nanostructures, DNA origami stands out, being a widely popular technique. For this reason, in Vivern, we focus exclusively on this design methodology, or more precisely, we focus on multi-layer DNA origami. As for the DNA origami itself, it assembles the structure using a long DNA scaffold strand, which is accompanied by multiple shorter staple strands. Together, these strands assemble the whole structure. As you can see, the staple strands are designed to be complementary to the scaffold on the predefined locations, ensuring that the structure will be well defined. Currently, there are various approaches to the visualization and design of DNA origami structures. 2D layout, while efficient for some tasks, can easily become too cluttered, and it might be difficult to imagine how is the structure supposed to look in 3D view. Solves this issue, naturally providing different perspectives on the visualized content. On the other hand, visualizing 3D structure using a regular desktop screen still presents some challenges, such as insufficient depth perception, as you can see in this example. After identifying the challenges and drawbacks of the current related work, we have decided to make a move from traditional 2D desktop interfaces to head-mounted virtual reality, since this medium was shown to be beneficial in aspects of natural visual exploration and interaction with spatial content. And these are exactly the topics where we saw drawbacks in desktop solutions for DNA origami. In this regard, Vivern proposes a range of visualization and interaction concepts, such as multi-scale visualization, domain-specific color schemes, focus plus context techniques, design rules constrained by underlying DNA origami paradigm, various modeling techniques, and finally, support for assembly of multi-structure complexes. In summary, the features of Vivern allow users to design or create various structures, either starting from existing structures or creating new ones from scratch, or to assemble completely new multi-structure complexes, including also proteins. In the following slides, I will briefly introduce some of the concepts, followed by a short demonstration of the application. During the design of DNA origami structures, information on various levels of abstraction may need to be considered. Therefore, in Vivern, we propose three distinct visual scales. First one, so-called nucleotide scale, highlights the individual nucleotides, their locations, and nucleobase types. This scale can be considered as a starting point, since thanks to the constraints of DNA origami, the atomic details might be mostly omitted. Second scale focuses on individual single strands, enabling to better perceive their routing, which is a crucial property for stability of the structure. Finally, double strand scale visualizes individual double strands, providing clearer overview of the shape of the structure and its core geometry. Individual visualizations can be colored in one of four available color schemes: nucleotide color scheme, color coding nucleobase types, scaffold scheme emphasizing the DNA origami scaffold strand, then single-stranded scheme, which distinguishes individual DNA strands, and finally a color scheme highlighting crossovers, an important architectural property of DNA origami structures. As for the focus plus context techniques, Vivern offers so-called magic scalens, enabling users to embed selected visual scale into another one, as shown on the slides. This technique allows for creation of customized visual representations, showing detailed view only when necessary. So, for example, in this case, the user starts with two double strands visualized in double-stranded scale, then he embeds single-stranded visualization into the double-stranded one, and then he continues by embedding nucleotide representation into the single-stranded one. Similar to magic scalens, Vivern also provides additional focus plus context technique called DNA twister. This feature enables to modify the visual scales by removing helical twist to better perceive the routing of individual single strands. Finally, additional interesting modeling concept employed by Vivern is the lattice constraint design. It's based on the fact that multilayer DNA origami structures are usually constrained to a particular lattice layout, determining positions where the structural elements can be created. Vivern preserves these constraints for each of the loaded structures. However, it allows for simultaneous visualization and modification of multiple structures at the same time, with each one located in its dedicated lattice. In this way, the internal constraints are preserved while the user is given the freedom to explore multi-structure assemblies. Finally, let's show a short demonstration of Vivern in action. In this video, we can see a user who loaded a structure from a Cardano file. Cardano is an application which is commonly used for design of DNA origami structures, and Vivern provides support for direct import of the JSON files from Cardano. As you can see, the user tries different visual representations and color schemes of Vivern as he continues to explore the structure directly in VR. The interactions with the structure are quite simple because the user can just grab the structure and move it around. After the exploration is done, the user tries to create a new double strand. As you can see, the double strand was created by default in double-stranded representation, and the user decides to use the Magic Scale Lens tool to change the representation of this double strand to a single-stranded scale 1. What you could have also noticed, the user disabled the visualization of the lattice and the wireframe to make it easier to see the structural data. Finally, when the new double strand was created, the user decides to use the break operation, which basically breaks a DNA strand and enables to connect it with another one, resulting in a creation of so-called crossover. After this operation is performed, the user can use Magic Scale Lens again to explore some locations in the structure using the nucleotide scale. The focus plus context techniques in Vivern work in two ways. You can either preview the location or change it permanently. Finally, the user uses another focus plus context tool called DNA Untwister, which allows to remove the helical twist. Apart from loading of existing structures, Vivern also provides a possibility to create structures from scratch. In this example, the user created a honeycomb lattice pre-filled with double strands, and he changed the representation of the structures to single-stranded scale. As you can see, the user can try different representations again, and then in the double-strand representation, he can remove the whole double strands from the middle of the structure to create a void inside. Then the user decides to create another lattice using the square layout and with two double strands. And again, the user can naturally grab the lattice or the structure, change the representation or other properties, and then he can move these structures and place them as he wishes. So he can again enable the wireframes to have more precise controls. And then basically he's free to move the structures around while keeping their inner constraints valid. So in summary, in this talk, I have introduced a virtual reality tool called Vivern for visualization and modeling of DNA origami nanostructures. Vivern offers visual representations adapted for the corresponding modeling tasks, focus plus context techniques naturally suited for virtual environment, and modeling interactions constrained by the underlying biological data. We also conducted a user study with 7 domain experts, and for the results of this study, I will refer you to the corresponding paper. As for the future work, we are mainly considering to explore analysis of coarse-grained molecular dynamics of DNA origami structures, as well as additional use cases for application of virtual reality in the domain of DNA origami. So that's it for this presentation. Thank you very much for your attention. At the bottom of this slide, you can find my email, and I will be happy to get in touch with you. Moreover, the slide also references again the TVCG publication this presentation is based on. Have a nice day.
So, and we have David here. Can you hear us?
Hello. I hope you can hear me.
Perfect, yes.
Great.
So we have a question. In the demonstration, the user is often switching between interaction panel and 3D structure. Attach the menu panel to point the device to avoid always moving back and forth. I guess the question is, would that be a good idea to attach the panel to the pointing device?
Yeah, for sure. Yeah, I agree that in the version we show in the video, basically, the user experience was not top-notch, actually. Yeah, so this would be definitely an improvement. I mean, one issue is that what we noticed that, for example, the biologists, they sometimes have an issue that if the menu appears directly in the 3D structure, then that's the problem. They don't know how to deal with that. But some way of attaching and overlaying the menu on top of everything what is shown would be definitely good in this regard. So I agree. And thank you for bringing this point.
Okay, and we have another anonymous question. Do you have any plans or current implementation for exporting 3D or 2D files for use in papers and publications? Other than just making a screenshot, I guess.
Basically, currently we have so-called screenshot mode. It was used in some of the presentation slides where there is this white background. So this is, let's say, small improvement or small feature to make the screenshots more easier. But there is no possibility to export 2D files, or I'm not sure if you mean screenshot by the 2D file or if you mean 2D design of the structure. That's a question. But I think this could be a potential interesting use case. I mean, if you'd be interested in this, you can also write me on the email in the presentation, and you can expand on the comment, or you can expand also on the Discord, I think. But yeah.
Okay. Unfortunately, in the interest of time, we have to move on already. So thanks again for joining, David. And I forgot to mention, if you have any or more questions for any of the speakers, please don't hesitate to also post them on Discord. And I think the Slido questions that were not answered are moved to Discord. So also to all the speakers, please have a look at Discord later on. So, okay.
Also, thank you and bye.
Bye, thank you. All right.
I would like to use it. Okay. Can you?
Good. That looks good. Okay. So our next speaker is Aditya Pandey, and he will talk about GenoRec, a recommendation system for interactive genomics data visualization. So, stage is yours.
Thank you. Good afternoon, everyone. I'm Aditya Pandey. I'm currently working with Regeneron Genetics Center, and I'm going to present the work that I did as a part of my PhD at Northeastern in collaboration with Harvard University. So DNA is a molecule that exists in our body which contains genetic traits, and a collection of all these molecules forms a genome. Genomes are important because their analysis has led to very interesting drug discoveries, and we all know how important that is, especially in the age that we're living right now. One of the most fascinating things that I think about genomics is the number of visualization, or the visualization design space it has. Just to set a context for this talk and present a motivation for my work, let me just go through a few examples. So in this example, we're showing a gene annotation plot which shows all the genes on a genome. In the second example, we can see a bar chart where the task could be to look at the correlation at different points in the genome. And finally, just to set more context and to scare everyone off, I wanted to show this third example, which is a complex overview of genome with these network arcs showing movement of data across different parts. Now, when we look at this, one question that arose in my mind, and when we talked to genomics analysts, was how did they decide which visualization to choose? And we learned that it was hard for them, and that makes sense because we know as visualization community, we spent years learning and perfecting the design space and the design decision of choosing a visualization. However, genomics analysts, unfortunately, don't have that kind of bandwidth. It would be good if we had some kind of solution or a tool that could recommend them or tell them what visualization to use. But unfortunately, in genomics, that kind of solution still doesn't exist. So that was our Eureka moment, and we realized, how about we build this tool? We build a system where people can come in, provide their data and task description, and get a visualization. But of course, it wasn't that easy. We had to break up this problem into smaller parts. So the first part that we want to deal with was to build a recommendation model that can take in data and task of the user and generate an interactive visualization. Being strongly in favor of having tools to support people, we also wanted to build a web application where users can come and provide this input and get the visualization output. And finally, we know that all of this would be of very little use if we didn't have solid evaluation to know if there is any value in the recommendation we're making. So in today's talk, I'll first very briefly go through our web application. Then I'll dive into the recommendation model a little bit and talk about our evaluation and show you some interesting insights. So I'll just be showing a video and talking over it. Please pardon me if it's a little bit too fast. I had to fit it in the time frame. So here we go. So GenoRec has this user interface where people can come and select the data sets they're interested in. These files are common genomics files. Based on the data types that they have, users can configure the information in these files. So here I'm just showing a demonstration of doing that. And once they do that, they can hit the Show Recommendation button in the recommendation panel, and GenoRec starts showing design variations which it feels it's appropriate. Now, one of the key aspects of GenoRec is that we allow people to make progressive changes. So people can go and keep on adding and removing files and see how that affects their visualization. So in this case, another file has been added, and when the user hits on the Show Recommendation, because it was quantitative data, so the circular plot was removed because it's a little bit less effective to see quantitative data with a circular plot. A key feature of our tool is that we also allow task selection. Now, in order to support that, we surveyed common tasks, and just to show an example, here I'm showing a compare multiple regions of interest task. And when people hit that, again, the recommendation system updates the visualization. And in this case, because the task is to compare between two different regions, we show this visualization with two views and this overview panel that can allow users to change between the different genomic regions. The final feature that we learned was we wanted people to have a feature to export their visualizations, because there's often a need for people to customize things at the end. So for that, we allow people to get this Gosling spec of the visualization that they're seeing, and then they can go and customize it. And Gosling is a genomics visualization editing tool. So now that we have seen the tool, now I just want to get into the details of what magic happens when we hit the Show Recommendation button. So in order to build a recommendation model, the first thing I had to think of was what are the visualization output in the system? What would be the visualization output? So genomics visualization, of course the answer was genomics visualization, but when we really looked at it, we saw that there's individual components that actually make up these visualizations. So for instance, in the circular plot, we have a saturation-based encoding, then we use a circular layout, and these circles are arranged in a parallel or a concentric way to create the final visualization. Fortunately, we had this information available by a survey, from a survey by Nusrat et al., where they identified the taxonomy of visualization. I would strongly recommend for you to check out this paper if you haven't. And using this information, we had to still figure out an order in which these steps should be carried out to create the visualization. And this step was important because we learned that there are some dependencies. So for example, the layout step was supposed to precede the arrangement step in order to create the visualization. Now, once we figured out the main steps or the components of the visualization, we had to build the recommendation knowledge so that we can translate this, the user input into the output. In our system, we use these decision matrix-based approach where the input that's shown in the columns is being used, the input that's shown in the columns and the outputs are being matched. So for instance, this matrix is showing that it's showing binary information where it's trying to show that the quantitative data could be displayed using a position or a saturation-based channel. And all this information that we use, we are actually dependent on literature that has been produced in prior to this work in information visualization. So once we had built the knowledge for all the steps, what the system does is that based on the input of the user, it tries to go through all the steps and identifies the most similar thing that it feels would be the best thing given the input. And then the rendering engine combines these things to produce the final interactive data visualization. So this was a very brief overview of the system. I would definitely recommend you to go and check out the system and read more about it. But now I'm going to dive into the user study that we did. So we brainstormed a lot that how do we evaluate the system, because we had so many different components to evaluate. I'm going to just talk about how we evaluated the utility of the recommended visualization, because this was one of the main focus and the novel aspects of the system. So in this case, we recruited genomics analysts who were actively working in the field, and we conducted a within-subject study where we gave them data and task combination phrased in their domain language, and then we showed them GenoRec stop recommendation and a median recommendation scored by GenoRec. So GenoRec can actually give you multiple recommendations, and some of the recommendations are not scored as much as the top recommendations. Of course, the participants did not know these stimuli were anonymized. And then we asked them to answer this question if they would use this visualization given the data and task description, and they could rate this on a 1 to 5 scale. So we did the study with 13 participants, and we learned that GenoRec did get higher utility ratings. However, I actually want to go into the things that did not work for GenoRec, because as scientists, I think we are more interested in what did not work. So I wanted to just highlight 2 main scenarios. These 2 scenarios, which you can read more about them in the paper. Sorry. So in these scenarios, GenoRec did equally or not as good as the alternate recommendation. So we tried to understand why that happened, and we saw that GenoRec, it was an overview task, and GenoRec was showing the entire genomic information in one view. However, when we talked to the analysts, they were actually preferring a view that splits up data by chromosome, because even for an overview task, they like the granularity, and they like to see granularity at individual chromosome level, which I feel could be useful if you're trying to design genomics visualizations. So I'm just going to talk about some takeaways from this work. So I've already discussed that the primary goal was to help genomics analysts get final interactive visualizations. However, I think there's a secondary goal which could be very important with recommendation systems, especially this one, is that we visualization designers could use these recommendations as an intermediate visualization design option and build over it. So because this is a recommendation system, we also want to extrapolate some of the findings that we had for our system, and if they could be utilized for future visualization recommendation systems. So one of the things that we learned during our interviews with our genomics analysts is when we recommended them design variations, they preferred and they liked it because they could see some options that they were familiar with, and there's prior literature in recommendation systems that familiarity actually leads to more trust in the recommended outputs. And the final outcome that we want to highlight is when we are recommending these visualizations, we want to focus on allowing users to customize, because visualizations do have a lot of design aspects related to them, and we wanted to just make sure that people can take our recommendation and customize it and use it in their own use cases. So with that, I'd like to conclude and thank the funding agencies, Harvard and Northeastern, for allowing me to pursue this research. You can learn more about my work on this using this QR code. And I want to thank all of you, because I know there's lots of parallel sessions happening, and you chose to come here, so thank you so much for that. Thank you.
Thank you. We are a bit behind time, but I think we have time for at least 1 or 2 quick questions. So Anonymous asks on Slido, what does the Gaussian spec look like for the exports? In other words, how can I, as an analyst, use the system in my workflow most easy to make visualizations with my own data?
That's a great question. Sehi would be very happy. Sehi built this Gosling specification. It's a JSON-based specification language, so you get the output in a JSON format.
Okay. And as someone who also teaches visualization and visualization design to bioinformaticians, biologists, I was wondering, does your system or have you thought about explaining the choices of the system to the users, so to educate them on the go while they are using the system, so to say?
Unfortunately, it doesn't do that explicitly, but we were hoping with the progressive configuration, it was somehow possible implicitly for people to know why, or try to understand why these changes were happening. But I think that's a great future direction, which we actually have discussed and we want to explore further.
Okay, there's another one. Were there any specific visualization types that genomic experts tend to prefer that a visualization expert would disagree with?
Someone who's working already, I think circus plot is something that has the community's genomics domain very divided. People think it's really cool, but there's actually research and information visualization which shows that reading data off circular tracks is hard. So That's something that I have learned, that it's a debatable visualization design choice.
And last very quick question from Anders Innermann. Can you specify multiple scales of interest?
I'm sorry.
Can you specify multiple scales of interest?
Explicitly not in the system right now, but I think with Gosling you could go ahead and change that information if you wanted to.
Okay. So yeah, thank you.
Thank you, everyone.
So our next speaker is Pavel Ulbrich, and I'm probably mispronouncing not only his name, but also the name of his paper, Small Boxes. Is that how? Yeah, perfect. Data flow model for molecular dynamics exploration.
Okay, so is it on? Can you hear me? Great. I'll just put down my mask and play the presentation.
Good.
Yeah, so thank you very much for coming to this talk or watching this online. My name is Pavel Ulbrich, and on behalf of my colleagues from Maastricht University of Brno, Czech Republic, from TU Wien of Austria, and from Lockschmidt Laboratories, also from Brno, Czech Republic, I would like to present to you our approach of visual exploration and visual analysis of molecular dynamics, a tool that we call small boxes. So this is a molecular dynamic. Simply said, is a computational simulation of movement and changes of all the positions of all the atoms within a molecule in time. And why do we analyze it is because it includes a lot of meaningful data. For example, the bonding of another bodies within the simulation as a ligand that we see in the front to the specific part inside the molecule, which we call active site. Or it can show us different conformations of the molecule, which may imply difference in functions and so on. So the problem with the molecular dynamics is that with the current power that we possess, they are reaching thousands of hundreds of thousands of snapshots. And by mere observation on a video, as we see right now, is not feasible anymore, as it would take days or even weeks to finish. So we need to have another approaches. And currently the domain experts, especially protein engineers, are focusing on specific parts as atoms, amino acids, or even the whole chains on the molecules. Also on the active side and ligands, as I mentioned before, which might bond eventually. And they are using several methods and descriptive functions to describe what is going on, such as distance, root mean square deviation, or even more computationally costly ones, as energy, bottleneck throughput, and so on. So by combining all of this together, they can have a pretty good overview of what's going on with a very long and very large computational molecular dynamic. So let me illustrate this on the example from some of the colleagues from Lockschmidt Laboratories when they were trying to describe the catalytic cycle of a specific molecule. So they started by the selection of several input structures, calculating the distance. Based on the distance, they calculated the energy necessary for traversing the distance, and so on and so on and so on. So they used a bunch of scripts to generate a lot of plots, which they put into superposition. They juxtapose them. Eventually, they had a pretty clear idea what is going on with the molecular dynamic. However, as you can see, that this approach may imply some problems. For example, hard application of these scripts into specific problems, hard hypothesis generation. And for the trained eye of domain experts, it loses the connection to the 3D. So in our previous work, we tried to mitigate this by introducing a set of descriptive functions, which we combine into what we called an importance function that would change the speed of the playback of the 3D video and would show these results on the 3D. However, when we were evaluating our method, it became obvious that the mental workload for the domain experts is too much. And we need a better way how to combine all the functions together. So we got back into our drawing boards and we come up with an application that is called Smallboxes. And in this application, there is always present 3D view on the left. Then on the bottom, there is a detail view that utilizes the whole width of the screen that the domain expert is using. And on the right, we can see a series of node instances that we call Smallboxes. They are connected together to form a great descriptive function for the molecular dynamics. So let me start with the description of a tool that we designed specifically for the long molecular dynamics. And apart from the traditional UI elements such as repositioning, resizing, minimizing, and so on, we also designed the specific connection links and pins. So we can maximize the use of these Smallboxes, and we can use only meaningful connections for the definition of the descriptive function. Apart from that, every Smallbox contains a line chart briefly describing the trends in the time series data and settings that are unique to each of the Smallboxes. So during the quite lengthy process of designing these Smallboxes, we come up with 3 main categories. And the first one is the input, as we can see on the left. In this case, this is atom, which was selected directly from the 3D structure. And what is immediately visible after selection is an approximate RMSD or root mean square deviation, which shows us how much this atom got away from the initial position of the simulation. The second main category are the measure Smallboxes, which already require a connection to the input Smallboxes, as distance in this example requires 2 input Smallboxes to calculate the ingredients distance in time between them. And as you can imagine, Smallboxes are rather small. That's why they are called Smallboxes. So we cannot really fit the whole duration of the time series into it. So we need to bin that. And that's the reason why we included the functional box plot and also the labels. So we see an immediate overview of the ranges that we are working with. The last category are the aggregation Smallboxes, which do not, as a maximum Smallbox in this case, and they do not introduce any new color, whereas they are reusing the colors of the distance Smallboxes, and they are using the connections to the input Smallboxes. So we see on the hierarchy in the canvas the immediate provenance of which measures, in fact, influenced the final descriptive function the most, and in which cases. Yeah. To connect the 3 views that I talked about before, we are using the snapshot bar, which is permanent in the measure and aggregation Smallboxes, and it sets the timestamp on the snapshot that we would like to explore in more detail. And one more thing is that we include a series of modifiers such as normalize, inverse function, and a derivative for better combinations of descriptive functions and inputs into the things. Well, I think I said quite a lot of things during this slide, so let me just recap it very quickly. We have input Smallboxes such as atom, residue, chain, or some subsections as an active site. We have measure Smallboxes as a distance, angle, or root mean square deviation, or computationally more expensive that we can load as a CSV. And we have aggregation Smallboxes as, as maximum, minimum, and average. Very quickly, the last challenge that we are trying to face now is more technical one, and is that typically molecular dynamics are in the tens of gigabytes. So we are outside of the real-time interaction realm. So we are using the principles of progressive analytics to very quickly calculate the first few iterations on the lower resolutions and to refine our descriptive function over the time, which can easily take seconds or even minutes for thousands of hundreds of snapshots. Let me quickly demonstrate the case on the video where we are comparing the one ligand in the simulation to the series of pre-computed values. And we would like to find out whether it entered or did not the cavity and what was happening while he was there. So we can load the throughput of the cavity of the protein, and we can compare it to the flexibility of the ligand as we computed it with RMSD. And of course, this is not enough, so we can very quickly introduce the input which is the bottleneck of all the residues that are forming the cavity. And we can calculate the distance between the ligand and these two. And once we'll normalize it, we'll get a very nice descriptive function, which will happen anytime soon, I guess. Yeah. So yeah, this is it. And if we'll connect it to the aggregation function, of course it doesn't want, but after normalizing, we can see very clearly that there is a plateau in the middle, and at the beginning it was different. So yeah, for the real use case, it's more complex, and due to the time restriction, I'm not going to talk about it, but I encourage you to search for it in my paper. There are two interesting use cases for the binding of 3 ligands and the protein unfolding. And very quickly, the future work, there will be more small boxes, there will be a bunch execution for multiple molecular dynamics, and we plan to release our software with the Caver Analysis 3.0. Thank you very much for your attention. I will gladly answer any questions that will be asked. And in the meantime, we can also see the video from one of our use cases. Thanks.
Okay, thank you. There are currently no questions on Slido, but I, of course, have tons of questions.
Of course.
Very interesting system. Since you kind of hinted at that in the future work, extending that with new methods, I mean, measures for the small boxes, how easy would it be to extend that system as a user? I mean, does it offer a plugin system or something like that where users could easily...
Yeah, right now, the functions that are computed within the small boxes in parallel, they are treated as modules. So what we plan for the future work is to extend it to, of course, include more modules and more functions. But we also plan to include as a custom-based small box, which would mean that the traditional approach with the scripts that the domain experts are using right now can be also used with this approach. So yeah, I think it's pretty scalable in this sense.
Okay, thank you. I also wondered, I mean, in the spirit of the last talk that we saw, do you think it would be possible, or do you even have something like that in mind already, to have a recommendation system for the 3D visualization, to adapt that to the parameters defined by the small boxes?
That's an excellent question, in fact. And we're thinking about it from the very beginning, that we can even track the interactions with the system of the domain experts. And based on this input, we can create a model. We can train a model that will recommend possible small boxes that will be useful in this situation and this specific molecular dynamics. So yes, of course, we have it planned. It's not implemented right now. We will be very happy to implement it in the future. But it's another completely new task, I guess, related to this one, of course.
Next year's Swiss paper. Okay, all right, then,
Okay.
yeah, let's thank Pavel again.
Yeah, thank you very much.
To the next talk. So our next speaker, while he's setting up, I can already announce him, is Andrzej Start. And again, sorry for mispronouncing your name. The paper title is Molecumentary, Adaptable Narrated Documentaries Using Molecular Visualization. So we are staying with molecular structures here. So Andrzej, stage is yours.
Okay, so good afternoon. My name is Ondřej Strnad. I'm from KAUST, and today I will be presenting our fully automatic technique for generating guided tours of molecular models. We call these guided tours Molecumentary, which stands for Molecular Documentary. I would like to highlight that this was a wonderful collaboration between Masaryk University, KAUST, TU Vienna, and INRIA. The problem we are addressing is something we encountered ourselves in previous projects. where we showcase these large-scale molecular models, as this HIV in blood plasma, to general audience. But general audience without expert description cannot simply understand the model, because they might not know which part of the model is important and which is not important. Therefore, for general audience, this might be only a sequence of nice pictures. From this perspective, there are these two distinct types of media where we can learn about biology. First are, of course, books. There are plenty of books, very nicely written in an engaging way, where we can learn about biology. However, the limitation is obvious. They cannot contain dynamics. The limitations are only to static images. The second media type are these visualizations. Still more and more people learn about biology from visualization and animations that are publicly available online. However, in this case, the production of these animations can be time-consuming and maybe even expensive. To address these challenges, we propose our conceptual framework of adaptable documentaries, when we propose changing the rigid parts of documentaries with something which is more adaptable and or even more scalable. So to be more specific, we propose using a real-time visualization instead of rendered animation, and automated exploration instead of auto transition. Because we wish to couple our visualization with commentary, with a voiceover, and we want to make it scalable, we cannot use pre-recorded voiceover. Therefore, we wish to use synthetic commentary. This is the overview of our framework. It consists of two main steps. Story graph foraging, where the story graph that contains the information about biological model is created. And in the, here. And in the second step, the story graph is traversed and narration and narrative is created. I will go through these two steps in more details now. And I will start with story graph foraging. So the input is a biological model that contains a hierarchy. And every type in that model is labeled, is named, it has its own name. So for every type, we create a node in the skeleton of story graph, and we copy the parent-child relation from the hierarchy. In the next step, we wish to associate with every node the textual description. There are multiple strategies how this can be done. We implemented two different strategies. One is to get the information from publicly available resources, such as Wikipedia. And if we cannot find the respective information for that given type online, we can provide additional textual description locally. So far, we have a story graph with structure edges only. We want to add even the functional edges. Again, there are multiple strategies how this can be done. For example, we can use something which is called metabolic pathways that describe the chemical reaction within a cell. But we decided to implement something easier. So, for example, this is a snippet of the text we got in the previous step, and by simply keyword matching, we can see that in one sentence, a capsid protein and RNA were identified. So therefore, we create a functional edge in the story graph. So with this story graph, we can continue to actually create the one narrative. And this is done by traversing the story graph, creating an order of nodes, and every node then is associated with a scene. These scenes are added to a timeline, and this forms the final narration. We have three different scenes. Focus scene explains the focused element. Overview scene presents the composition of the focused element, and transition scene is used for transition between focus and overview scene. Every scene has its visual and audio part. In the visual part, we implement the procedural camera animation with three types of animations and advanced visibility management to highlight focused elements. And this is done by implementing a traveling cutting plane approach with the possibility of extending focused objects. In the audio part, we have three different types of commentaries. First is descriptive, that provides the full detailed information about focused elements. And then we have structural commentary that describes the structural element, and navigational commentary that acts as a filling material, as connective material. Structural commentary and navigational commentary are based on templating approach, where we specified a list of keywords that are evaluated in the real time. We have two different variants of mockumentaries. First, we call this self-guided, where the narrative is stochastically determined in real time. This provides us with the possibility to generate different mockumentaries basically with every run of the system. So you can see that we started from the very same position, but immediately the first node was chosen differently, and then the mockumentary continues. The second type of mockumentary we call is text-to-mockumentary, where the input is the text with that contains the predefined keywords, and this basically defines the narrative.
Researchers are studying GP120 in the search for new approaches to creating an anti-HIV vaccine. Several viral enzymes are packaged inside the virus. Reverse transcriptase will create a DNA copy of the genome when the virus infects a cell. Integrase will splice this DNA copy into the cell's own genome, which then forces the cell to make many copies of the viral proteins.
So this work was a step towards fully automated storytelling. From our perspective, it was interesting to couple all the audio narration, labels, subtitles, and visualization together, which is not used for very used for documentaries. Our goal at the very beginning was to exploit the data, the text that is already available online, and I believe we managed to do that. And that's, I think, all what I wanted to say. Only one remark, we already have a follow-up paper tomorrow, which is called Nanotilus, and will be presented by Ruwaida, so please check that out too, and thank you for your attention.
Yeah, thank you. So in the intro video that you showed, that was manually created, there was also this temporal component, so it's animations of this documentations. And if I understand you correctly, so far you're just working with static data, right? So do you have any thoughts on how a time component, either using a manually created animation or some simulated materials, would integrate into your system?
So our system basically requires two inputs. One input is a model with hierarchy, and for example, a text. The text can be created by an expert biology user. For example, a lecturer or a professor who wants to show a narration, who wants to show a story, but he doesn't necessarily have the skill to program. So if we have these two inputs, we can directly plug them in our system and we can get the resulting visualization, the molecumentary. So, yeah, did I answer your question?
So it would be pretty straightforward.
Yeah,
so. Okay.
Yeah.
We have another question by Mark Keller. Where does the 3D structure come from? Is it available of 3D models? Is the availability of 3D models a bottleneck in this approach?
Currently, I see that as a bottleneck, yes, but these models are being created, especially in our group, because we are working on a system that makes the generation of this model much easier and more available for general audience.
And Marty Hurst asks, or comments, this is really cool work. Have you considered adding text annotations to point out what each component is, e.g. what molecule is what or where connections are happening?
Sorry, can you please repeat the question?
Have you considered adding text annotations to point out what each component is? For example, what molecule is what, or where connections are happening?
Yes, I think this might be contained as the text. So, of course, if you really insist on having some kind of information in the system, so either you need to type it manually, or there is a vocabulary basically of these template messages or template sentences. So you might add these into the templates and then the system will work with that.
Okay. And one last quick question by Anders Innermann. What happens if you in the text refer to something that doesn't exist in the visualization? What does the system do?
Yeah, so the model comes with hierarchy and with a list of elements, with types of elements. So if you refer in a text to element which is not in the model, it just omitted, it's skipped.
Okay, all right. Good, then let's thank Andrzej again. And let's move on to the last talk of this session. It's again a virtual one, but the speaker is also here on Zoom for a Q&A afterwards. So even if we are running a bit late, please bear with us here and stay for the Q&A. So the last talk will be given by Furui Cheng, Polyphony and Interactive Transfer Learning Framework for Single-Cell Data Analysis. Should come up any second. Awesome.
My name is Furui Cheng. I'm a PhD candidate at the Hong Kong University of Science and Technology. Today, let me introduce our paper collaborated with Harvard Medical School. The paper title is Polyphony, an Interactive Transfer Learning Framework for Single-Cell Data Analysis. We know that cells are the basic unit of life, and many challenging biological questions are about cells. These years, single-cell sequencing techniques have been increasingly developed to answer these questions by examining the sequence information from individual cells. And when analyzing the single-cell gene expression data, the first challenge is to annotating the cell type, which is usually done manually and is time-consuming. So recently, biologists have been getting more interested in transforming the labels from a well-annotated dataset to newly produced data, which is more efficient and reproducible. And the problem sounds like a table classification problem that can be resolved with any traditional supervised machine learning method. However, it is not that simple. If we project the cell gene expression data to a 2D space, we will find that the reference and core cells are not aligned well. This is because each experiment is conducted under different conditions, using different sequencing techniques. The results of each study are systematically biased. And technical variations are often mixed with true biological differences, like the existence of novel cell types or states, which should be reserved. We want to remove the technical variations but keep the biological differences, and it isn't easy. Existing computational approaches remove the batch effects by mapping the core cells to the most similar reference clusters, but it not always works. The model may undercorrect or overcorrect the dataset in some cases. And the annotations made by the biologists are more robust. And when biologists validate the annotation results, they will consider more aspects, like the marker genes in the cell clusters and the genes' functionalities, interactions, and pathways. This external knowledge is very unlikely to be represented in the training data. Besides, cell annotation is not only a computational problem. Experts want to know what is inside their data and gain valuable insights. Traditionally, they use visualization to explore the data interactively, but existing visualizations are not designed for reference-based annotation. So how can we bridge the visualization approaches and the computational annotation methods and keep humans in the loop? We first interviewed several molecular biologists who are experts in single-cell data analysis, and we found that the experts analyze the data on 3 levels: dataset, cell population, and gene. And also, experts expect to understand the integration process and interact with the model for refinement. And according to these insights, we proposed a mixed initiative analysis workflow that incorporates experts' knowledge in an interactive and iterative integration process. The main idea is to use anchors to support communications between humans and the model. An anchor is a pair of analogous cell populations across datasets confirmed by users. And for example, the system recommends several pairs of cell populations that should be integrated, and users can validate the model recommendation to get the model to make reasonable predictions. Based on this idea, we contribute a paradigm for communications between users and the model, and an interactive visualization system to obtain user feedback. Then let me first introduce this framework. The framework contains 4 steps. In the first step, the system recommends anchors to users, and it is done by first building reference clusters and then assigning the most similar power cells to reference clusters. In the second step, users provide feedback by interacting with anchors and using the polyphony interface. In the third step, the system fine-tunes the model according to the confirmed anchors, where we try to simultaneously minimize the original loss and the distances between the confirmed anchors and their corresponding clusters. After that, the embedding of all power cells will be updated. And then let me introduce the visualization design. And we first project the cells from the latent space to a 2D plane with a UMAP algorithm. And the color encodes the cell types of the reference cells and predict the cell types of the core cells. To distinguish the reference cells and core cells, we use contour lines to encode the density of the reference cells. Then we use glyphs to annotate the anchors, and each glyph links the centers of the reference part and the core part. The Vis of the shadow encodes the anchor assignment uncertainty, calculated according to the similarity between the marker genes in the 2 parts. And a bold link indicates that the system is not confident about the anchor, and users may need to take further inspection. These designs help users to gain an overview of the joint embedding space and understand cell correspondence. The anchor set view organizes the anchors in a table, which helps users understand the anchor profiles. And gene-level understanding is supported in the marker view. When users select the anchor, we present the top differentially expressed genes in both core and reference cells. And we separate the shared top genes and the unique ones, and show each gene's z-index and ranking on the core and the reference sides to help users make comparisons. Here's a demo showing how users may interact with the system. The user inspects an interesting anchor composed of pancreas gamma cells and clicks the top marker gene to see how the gene expresses in anchor cells. The users use lasso to refine the cell selection. The users can also delete an unwanted anchor suggested model and also add a new one. After confirming several anchors, the users update the model and gets better integration results. Then let me introduce the evaluation of this research. We use two commonly used benchmark datasets, human pancreas dataset and a PBMC dataset. We evaluate the system from both the algorithm perspectives and also the human's perspectives. In the algorithm evaluation, quantify the effectiveness of the system with user simulations. This experiment aims to measure the improvements in the integration and annotation quality after using the system, and we adopt the commonly used LYSIS scores to assess the integration quality and use F1 scores to assess the annotation quality. All scores are normalized to 0 to 1, and a higher LYSIS score indicates that the batch effect is more thoroughly removed. A higher CLYSIS score indicates that different types of cells are better separated. We simulate the user confirmation actions, which are a subset of possible user operations. And we consider 3 selection strategies: selecting no anchor, which is a baseline, selecting all anchors, and selecting only the good ones. We leverage the ground truth cell type labels to assess the anchor qualities, and intuitively, the anchor has a good quality when the cell types in the query and the reference parts are consistent. And we use a threshold theta to denote an anchor selection policy, where a lower theta indicates a stricter strategy. Here are the results. You can see that all polyphony models with anchor confirmation outperform the baseline models. And we also noted that the best annotation performance comes from selective confirmation policies, which means that not all anchors help improve the annotation accuracy. So we still need experts to select the feasible anchors, which confirm our motivation. Then let me briefly summarize the contributions of this work in resolving domain challenges and improving their workflows. We improve the traditional workflow by joining two separately stages, integration and understanding together. We found that anchor-based methods leverage data correspondence for integration, and one of the major goals of integration is to help experts understand data correspondence. So why not join them together? And in our approach, we did the visualizations and interactions to transparentize the integration process. And thanks for listening. You can check the GitHub link for more details. Hello everyone, my name is Fred Cheng. I'm a PhD candidate at the Hong Kong University of Science and Technology. Today, let me introduce our...
So, good, we have Furu here. Can you hear us? Quick mic check.
Hello, can you hear me?
We can hear you. Can you hear us?
Yeah.
Very good. Okay. I was wondering, I mean, your visualizations that help users make an informed decision, did you evaluate or did you collect any feedback how well the users, the experts could work with that? I mean, how confident were they in the end in the decision they made?
Yeah, so actually we work with molecular biologists and conduct some qualitative studies and to see how they would like to use this system. And so you can see the papers for more details. So currently, so we find that we still need to train the experts or still need to further work with them to understand so how do they interpret this anchor and how would they, I mean, would like to make the judgment of whether to accept an anchor.
Okay. And is scalability with respect to markers an issue here, or does that not play a role in the system?
So currently our system is tested with the cells with about 100,000 cells, and so actually we find that for so each apple will takes about from several minutes to dozens of minutes, and but since I mean the batch effect removal, actually this task is quite challenging and usually it's time consuming, so this time is still considered as accessible by the users.
Okay, so computation time is, of course, one point here, but the visualization itself is scalable enough for the respected data set size. Okay, so that was not an issue. Okay, all right, thank you. I currently don't see any more questions on Slido. So I would say let's thank Furui and all our speakers again. And this concludes the session. Thank you all for coming, and thanks to everyone for giving their nice presentations. Thank you.